Hi, my name's Will Giammattei, and me and my commentator, Rowland, are here to talk about the matchup today between mitosis and meiosis.
This is an extremely close matchup. Both are out of the gates fast, and they both start with interphase. Interphase takes the longest. This is the part where both cells get ready to divide.
Also noted, there are 3 stages of interphase. The first one is gap 1 phase. During this stage, the cell needs to decide if it wants to divide.
Now the second part of interphase is the S phase. During S phase, the DNA replicates, and there are 2 strands of DNA.
The last part of interphase is the G2 phase. During this phase, the cell makes the final adjustments to either do mitosis or do meiosis.
Now both have entered the prophase stretch.
Chromatin, or genetic material, in each cell has come together into a very visible shape.
Looks like the chromatin have a tag team thing going on, Will.
Replicated chromatin pair up with their originals and form a single chromosome.
One might call them sister chromatins. I see meiosis using the sister chromatins play as well. But then the chromosomes of cells are pairing up as well.
It appears that they are forming homologous chromosomes and swapping genetic information.
Look at that crossing over. It appears that the centrosomes have made their move and are heading for opposite sides of the cell. Also, the mitotic spindle has come out from the centrosomes.
Centrosomes and spindles are extremely crucial in the movement of chromosomes.
Look, Will! The nuclear envelope has disappeared.
And now the mitosis cell has advanced to the next milestone. Prometaphase.
The spindle has now grabbed onto the chromatids.
Notice how each individual chromatid has its own microtubule partner to guide it.
Just how are those microtubules hanging on so well, Will?
It's a tricky little thing called a kinetochore. Each individual chromatid has one to attach itself to the microtubules.
In the other corner, we have meiosis. Still in prophase? I guess meiosis just doesn't go through prometaphase. It seems the same as mitosis prometaphase anyway.
Look closer, sports fans. There's a slight difference. Each individual chromosome from a homologous pair has its own kinetochore.
Interesting.
Whoa! It seems that mitosis has entered the metaphase stretch.
The chromosomes have lined up right in the middle of the cell.
If you turn your attention away from the center, you'll see that the 2 centrosomes are on complete opposite sides of the cell.
I noticed that the 2 spindles on a chromosome go to opposite sides of the spectrum.
Keep in mind that each chromatid in a pair has its own microtubule. The whole chromosome won't be going to the same side of the cell. Oh look, meiosis is also hit metaphase. The homologous chromosomes have lined up in the center.
Once again, the microtubules are going in different directions.
It's anaphase! The chromatids are now separate and running in opposite directions along the microtubules.
Will, look at meiosis! The homologous chromosomes have also split!
Holy cow! Meiosis is also an anaphase. The chromosomes are moving into opposite directions.
And just like that, it's over. Phew! Anaphase really is the shortest phase.
Oh, we're not done yet. It's time for the home stretch. Telophase and cytokinesis.
What do you think happens? 2 nuclear envelopes?
That's right, Andrew. Now that the chromatids are split, they're individual chromosomes. With 2 full sets of chromosomes, the cell needs 2 envelopes. You'll also see the chromosomes relax and become far less bundled up.
I guess all that leaves is cytokinesis.
Check out that cleavage furrow.
The whole cell has split in 2. There are now 2 identical cells. Mitosis has finished.
But wait, meiosis still has to finish. Unlike mitosis, meiosis has 2 divisions. After the meiosis cell divides into 2, both resulting cells still have an equal number of sister chromatids that need to split. Thus, the cell needs to get ready for another prophase.
A prophase 2? I noticed that the spindle has formed, and the sister chromatids are beginning to move.
Unlike mitosis, the sister chromatids are not genetically identical. You'll see that the microtubules have attached to the individual chromatids. There's another metaphase where the sister chromatids line up on the metaphase plate. During anaphase, 2 sister chromatids go toward the opposite sides of the cell. Then, during telophase 2, the 2 nuclear envelopes form. Now we'll see the cytoplasms divide again.
Then that would make 4 cells? Wow!
Yup, but there are major differences in these cells. None of them have the same genetic information, are completely different from the father cell.
Yeah, I also see the chromosomal count is different. Each cell only has half the regular number.
These aren't your everyday cells, Andrew. These are sex cells. They only need half the regular number to fulfill their purpose. This is called a haploid cell.
As opposed to regular diploid cells?
Yup.
Thanks everyone for watching today. From Cell Cycle Television, we say goodbye and good night.